Hey everybody, it's Mr. Bliss. This video is about DNA replication. If you have not already watched the videos for nucleic acids or on cell organelles and you think you might need a refresher, I would go back and watch those videos before this one. A lot of the terms, a lot of the vocabulary referenced in this video was originally defined in those. So go back and watch those and then give this one a shot. If you're still here, welcome. Let's talk about DNA replication. DNA replication is important because as multicellular organisms grow, they need to make sure that every single cell contains a complete copy of the DNA. Every single one of your cells contains the exact same DNA as every single other one. And that makes sense because you originally came from a single fertilized egg. And then your DNA replicated as your cells divided through the process of mitosis. So your skin cells and your nerves, all your neurons, all of those contain the same exact copies of your DNA. It's just kind of the genes in your DNA that are expressed differently that makes them different. But in order for each one of those cells to contain a copy of the DNA itself needs to copy first. And so let's look at this process. And first, let's check out, like a brief overview of what happens. On the left side of this diagram, you will see, of course, a DNA molecule. And in previous videos, we talked about how, although this is a DNA molecule, it's actually made from two different polymers, two different strands. And they kind of run in opposite directions. We call it running anti-parallel. And so the left side will be facing this way and the right side will be facing this way, or vice versa. And that's going to be important for how this actual process occurs. On the right side, you'll see that I've drawn the two sides of the molecules in different colors. And that's because when DNA replicates, the original molecule unzips, and each side acts as a template for enzymes to build a new side. So if you look over here, this purple side is going to represent what came from the original molecule, and this yellow side is going to represent the new strand of the DNA. And because half of the DNA is maintained or is conserved in this process of DNA replication, we call this process semi-conservative. And that is important. All right, so if we look at our cast of characters here, this process is actually going to be a little bit more complicated than what I'm describing here. But for the sake of my class, these are the processes, these are the enzymes that I'm going to ask my students to know. And so we have 4 enzymes here, and they each have a different job. And with all enzymes, with most enzymes at least, their functions we can easily discern from their names. So most enzymes end in ASE, in ACE. We have amylase, lipase, on all of those end with ASE, but the prefixes give us an idea of either what they do or what they act upon. So let's look at our first enzyme here, helicase. Well, we know that DNA is a double helix, a double spiral structure. So this enzyme is important for changing that structure. Specifically, helicase unzips the double helix. And that's going to give us a platform where the other enzymes can come in and do their jobs. Now, we're going to skip DNA polymerase for a second. We're going to come down here to RNA primase. And the reason why is because it is the next enzyme in the process, and it's going to set the stage for polymerase. So again, ASE, enzyme. And what it's going to do is it's going to act as a primer. It's going to prime the DNA for the DNA polymerase to act on it. And what it's going to do is it's going to lay down some RNA nucleotides. And the RNA nucleotides are going to act like a little bit of a flag and say to the DNA polymerase, hey, you can start doing your job here. So RNA serves as a marker or starting line, starting point for the DNA polymerase. Okay. All right, so we have DNA polymerase. It says DNA polymerase 3. There are other polymerases. This one is going to be important for this process. Don't worry about the others. And again, ASE, enzyme, and it is going to form a polymer. In previous videos, we talked about what a polymer is. It is a repeating sequence of subunits that combine into a single molecule. And so this particular polymer that it's going to make is DNA. And it's going to do that by snapping together nucleotides. You have nucleotides that are kind of freely floating in the nucleus, just like this, right? And the DNA polymerase is going to snatch them up and stick them into the right places, using one side of the DNA as the template for knowing which one goes where. Like we said before, we know that if the template says that it's an adenine, then the DNA polymerase needs to grab a thymine, because adenine always binds with thymine. And if it's a cytosine on the template, the polymerase is going to have to insert a guanine, because those are the bases that match. They're complementary to each other. So what does DNA polymerase do? It forms a polymer from free DNA nucleotides. It's building. That's what it does. It's a builder. And then our last enzyme here that we're going to need to know about is ligase. And what ligase does is it binds. Ooh, that's going to be too short. It binds together fragments of DNA. What we'll see here is during this process, we're going to have sections of DNA that are unconnected, and ligase is going to act as almost like a sort of glue to stitch them together. All right, so here is our molecule. You can kind of assume that our double helix has been straightened out into a nice straight ladder, you know, from this sort of process into like a straight ladder like this. And then also it's begun to unwind. And the reason why we've drawn it like that is because it would take too long to draw in all those changes. So already what we're seeing here is that our first enzyme, our helicase, which I made this color, is going down this molecule and unzipping the structure. It's just going down and unzipping it this way. The other thing that you'll notice is that we have these numbers. We have our 5' over here and our 3' over here. And then we have 3' over here and 5' over here. And if you remember from our nucleic acids video, all that means is that the sugars on our nucleotides are pointing in a particular direction. So they're pointing towards the 5' end. On the opposite side, right, our anti-parallel side, our sugars are actually going to be pointing in the opposite direction toward the 5' end. And so this just tells us which direction the nucleotides are pointing. Why is that important? Because our enzymes are going to be, some of our enzymes are only going to be working in a particular direction on the molecule, usually that 5' to 3' end. So as our helicase moves down the molecule, we are going to be ready for our next enzyme to come in, which we said was RNA primase. So our RNA primase is going to come in here, and it's going to lay down a short section of mRNA. And I'm just going to make them all the same base here in this nice pink because it's visible, that's all. Obviously, they would be normally matching these nucleotides with the correct bases, but just simplicity for the sake. However, on the opposite side, we're going to have an RNA primase come in, and it's going to do things a little bit differently because of that difference in directionality. It's actually going to lay down a few primers, and the reason why will become apparent momentarily. We said before that the next enzyme, the most important enzyme in this process, is DNA polymerase 3. So here's our DNA polymerase. It's going to come in. It's going to see our primer here. Remember, we said that's going to act like a little bit of a flag as a starting point. But DNA polymerase can only go in that 5' to 3' direction. So on the top, it's not going to have any problem. It's going to start laying down that DNA this way. Okay, and we'll just assume that our polymerase has already moved from the primer all the way down to this point, and it's put in all, our correct nucleotides. Obviously, I'm going to do this out of order so I don't have to go and spend, 20 minutes drawing it in. But in reality, that's not what goes together. All right, that's good enough. And so it's moved down and it's put our bases here, and it's moved in a nice, smooth, straight line from that 5' to 3'. So this top side of our molecule is going to be known as our leading strand because it goes just smoothly, quickly. However, in the opposite direction, if it needed to start at the 5' end, it would have to wait for our helicase to finish unzipping this whole molecule before it could even start, and that's kind of a waste. So what it's actually going to do is it's going to come in and it's going to find our primers here, and it's going to do it in these short bursts. Okay, and it's going to do it here as well, and it's going to go through and fill in as much as it can just in short bursts. And so it'll lay this section down like this, and then it'll lay this section down like this, or vice versa. And, same thing here. It's not going to be a pretty illustration, but hopefully you'll get the idea. That's good enough. Sure. And so it goes through and it lays down those DNA sections in these short fragments. And we actually call those Okazaki fragments. It's probably my favorite term in all of biology. I'm going to start a band someday called the Okazaki fragments. Please don't take that idea. It's $1 million idea. And so because this side of the DNA has to happen in short bursts, we are going to call this the lagging strand. Okay. On the top, we have the leading strand. On the bottom, we have the lagging strand. But you'll notice that these fragments are not all together yet. First, the DNA polymerase will come back through and it'll proofread and take out those RNA primers. But finally, we'll have that ligase come in and just stitch these sections together. And so we're going to end up with two complete identical molecules at the end of DNA that should look relatively like these two are semi-conservative, two identical DNA molecules. And that's DNA replication. If you have any questions, leave them in the comments and I can do my best to answer them. Thanks for watching.